Chapter 4 focuses on the importance of the atom carbon and the ways that carbon lends to so much molecular diversity in life. Remember, carbon is the sixth element on the periodic table of elements. This means it has 6 protons. As a quick insight, carbon is the second most abundant element in our body and is the 14th most abundant element on Earth. It is found everywhere from cereal and pasta to clothing, furniture, plastics, and the air we breathe. As you may know, carbon also makes up graphite, used in pencils, and, of course, diamonds. So how does one element find itself literally at the center of such vastly different molecules? That's what we will discover in this chapter. By the end of this chapter, you will be able to model and categorize the fundamentals of molecular structure and functions associated with living organisms. Specifically, you will be able to describe the importance of carbon and how it produces large, complex, and diverse molecules. And you will be able to describe the variety of functional groups that are key to the functioning of biological molecules. As I mentioned, carbon is the second most abundant element in our body. 18% of us is carbon. Of course, it never exists solely as a carbon atom. The carbon is attached to other prevalent atoms such as oxygen, hydrogen, and nitrogen to form molecules. And carbon tends to be the backbone of these molecules. In fact, carbon is unparalleled in its ability to form large, complex, and varied molecules. For example, all proteins, DNA, and carbohydrates are composed of carbon. Other examples include fatty acid chains that make up fats and cell membranes, steroid hormones like testosterone and estrogen, and carbon dioxide, which you release with each exhale. All of these and other compounds that contain carbon are called organic molecules. Organic chemistry is the study of carbon compounds. Organic compounds can range from simple molecules to colossal ones. It is clear that carbon-containing molecules such as proteins, carbohydrates, and DNA are crucial to all living things. Not only are they important to living things, they also tend to be made by living things. This led scientists to question how the first organic molecules, which would later become the building blocks of life, were originally created. Stanley Miller performed this classic experiment demonstrating the abiotic synthesis of organic compounds. Abiotic simply means it is not derived from living things. The experiment was simple. Miller began with seawater, which he boiled into a vapor. The vapor rose and traveled to an artificial atmosphere containing CH4, which is methane, NH3, which is ammonia, and H2, which is hydrogen gas. He then sent an electrical charge through the flask via an electrode. The vapor was cooled by a condenser, creating rainfall, and this rain was collected and analyzed for existence of organic molecules. Miller demonstrated that most of the amino acids, which are the 20 building blocks of proteins, can be synthesized under similar abiotic conditions. Energy sources such as lightning and radiation may have originally helped to catalyze these reactions. Interestingly, amino acids are found in meteorites and interplanetary dust particles. When they rain down through our atmosphere, they carry these building blocks of life, though not necessarily life itself. This experiment gives insight into what may have sparked the creation of those original building blocks of life. Now let's dive into how to build organic molecules with carbon. The most common element constituents of organic molecules are carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. They tend to be found in uniform percentages across all species. In other words, humans, dogs, and snails will all have similar percentages of CH, O, N, S, and P. Just from these 6 elements alone, we are able to build a plethora of organic, carbon-containing molecules. This versatility is all due to the unique bonding capability of carbon. The magic of carbon is due to one thing: its ability to make 4 covalent bonds. Of course, an atom's ability to bond is solely due to its electron configuration. So in order to fully understand and appreciate carbon's bonding ability, we have to take a closer look at the subatomic level. Let's do a quick review of carbon. This is called an electron distribution diagram. It depicts the electron configuration of carbon. Carbon's atomic number is 6, meaning it has 6 protons, and its mass number is 12, meaning it has 6 neutrons in addition to the 6 protons in the nucleus. To create a neutrally charged atom, if it has 6 positively charged protons, it would need 6 negatively charged electrons. We see the electrons pictured here in this model. We learned in Chapter 2 that each ring around the nucleus is called an energy shell. The first energy shell is filled with 2 electrons. The second energy shell gets filled with 8 electrons. Carbon has a total of 6. Therefore, 2 are in the first energy shell, and the remaining 4 are in the second energy shell. Keep in mind that the electrons do not orbit the nucleus in a fixed circle. This is a Bohr model of carbon, named after the Danish scientist Niels Bohr. Notice how the electrons move around the nucleus without a particular pattern in what we refer to as an electron cloud. This simulation does not capture how incredibly small and fast electrons are. When scientists study electron movement, the electrons are so difficult to track that they appear to hop into and out of existence. The most important electrons of an atom are the ones that are in the outermost shell, called the valence shell. These electrons are called valence electrons. These are the reactive parts of the atom. Remember, each energy shell wants to be completely filled. This concept is called the octet rule. Carbon already has 4, so it wants 4 more to be filled and satisfy its octet rule. How do atoms gain more electrons to fulfill their octet rule? If you guess by borrowing or stealing from another atom, you are correct. Whether an atom steals or shares electrons is dependent on the atom's electronegativity, which is an atom's affinity for electrons. Many things factor into what determines electronegativity, but you don't have to worry about that. Just keep in mind that fluorine and chlorine are highly electronegative, and therefore they tend to steal electrons and form ions. They then can form a special bond called an ionic bond, which is typically found in salt. For example, sodium chloride is Na, which is sodium, with Cl, which is chlorine. Oxygen and nitrogen are very electronegative, though less so than fluorine and chlorine. So they want to share electrons, forming covalent bonds. If oxygen or nitrogen bond with carbon or hydrogen, which have lower electronegativities, these will create a polarity where oxygen and nitrogen are hogging the electrons, and we call this a polar covalent bond. If atoms bond with other atoms that have similar electronegativities, we again form covalent bonds where they will be sharing electrons, but this will be a non-polar bond because there will be no polarity. So now we understand that carbon has 4 valence electrons and wants to fill its valence shell with 4 more electrons. To do this, it will form 4 covalent bonds. Each bond counts as a shared electron. Here are some examples. Methane is CH4. With this molecule, we see one central carbon with 4 single covalent bonds to hydrogens. Ethane is C2H6. When we build organic molecules, we always start with creating a chain of carbons. We see that in ethane, 2 central carbons attached with a single bond. Off each carbon, there are 3 single bonded hydrogens. Notice that the carbons each have 4 bonds emanating. Ethene, or ethylene, is a little different. Here we're introduced to double bonds. Notice that the 2 central carbons are now double bonded together. Each bond still provides the carbon with a shared electron. Each central carbon now has only 2 hydrogens emanating from it. Why do they have 2 bonded hydrogens each instead of 3 like in ethane and methane? If we bonded 3 hydrogens to a carbon, the carbon would now have 5 bonds, 2 from the double bond and 3 from the hydrogens. Carbon must always have 4 bonds. Now you understand the connectivity of carbon, I also want you to note the structure. Take a look at methane. When carbon has 4 single bonds emanating, the atoms orient themselves into the tetrahedron. The tetrahedron is the same shape as the triangular pyramids. It's a polyhedron that is the same shape and size on all 4 sides. Atoms like to orient themselves to be as far away from each other as possible to reduce what we call steric hindrance. The tetrahedron satisfies that. Now look at ethane and ethene. Very similar molecules with one major difference: the double bond between the 2 central carbons in ethene. Imagine if you're holding a ball and stick model of the ethane molecule. You'd be able to twist and rotate the 2 sides around the central carbon bond. The same is true of the actual molecule. To contrast this, imagine you're holding a ball and stick model of ethene. Could you freely rotate the right and left side around the central double bonds? Definitely not. You could bend it a little, but if you applied too much force, the bonds would break. The same is true of the ethene molecule. Also note that due to the nature of the double bond, all atoms in the ethene molecule are on the same plane. In other words, they lie flat on that surface shown. The atoms of ethane and methane are not planar because they're comprised of single bonds which can rotate freely around the axis. By now you should know how many valence electrons carbon has in its valence shell, 4, as well as how many electrons carbon needs to satisfy the octet rule. It needs 4 more. It will also serve you to set to memory the information for the 3 other most abundant elements in the body: hydrogen, oxygen, and nitrogen. You may have noticed the word valence pop up many times in this lesson, with each a slightly different meaning to it. Note the differences so as not to trip up during the exam. We have valence shell, which is the outermost shell, valence electrons, which are the electrons in the valence shell, and now we have just valence, which is defined as the number of covalent bonds an atom can form. Using carbon as an example, the valence shell is the second energy shell. There are 4 valence electrons, and the valence is 4. It can make 4 bonds. An atom's valence is determined by the number of unpaired electrons in the valence shell. And with carbon, we see there are 4 unpaired electrons. Let's look at hydrogen. Hydrogen's atomic number is 1. 1 proton will be in the nucleus. The mass number is also 1. You may not have noticed this, but the version of hydrogen that appears on your periodic table is an isotope of hydrogen with 0 neutrons. If hydrogen has 1 positively charged proton, it must also have 1 negatively charged electron for a neutral atom. This 1 electron is placed in the first energy level, which is also the valence shell in this case. How many electrons does the first energy level need to satisfy its octet rule? If you answered 2, you're correct. If you answered 8, just remember 8 electrons is for the second and third energy levels. So, for hydrogen, the valence shell is the first energy level. There is 1 valence electron, and the valence is 1. Take a moment to figure out the valence shell, valence electron number, and valence for oxygen and nitrogen. Oxygen's valence shell is the second shell. There are 6 valence electrons, and oxygen has a valence of 2, meaning it must form 2 bonds to satisfy the octet rule. Nitrogen's valence shell is also the second. There are 5 valence electrons, and nitrogen has a valence of 3, meaning it must form 3 bonds to satisfy the octet rule. Now we understand how electron configuration of atoms determines its ability to create bonds. Carbon has covalent compatibility with many elements, the most frequent being hydrogen, oxygen, and nitrogen, and of course, carbon itself. Two examples of carbon partnering with non-carbon partners are carbon dioxide and urea. You now have all the information needed to build the molecule urea. Take a minute to try. Keep in mind the valence for carbon, oxygen, nitrogen, and hydrogen. When you're done, look up the structure and compare what you've made. We've discussed the omnipresence of carbon, its role in organic chemistry, and the importance of its ability to form covalent bonds and build complex molecules. As we dive further into how to build organic molecules, it will become clear why earlier in the lecture, carbon was described as the backbone to life. Remember this rule when building molecules. Always start with the carbon, follow with the nitrogen, then the oxygen, and lastly, hydrogen. The first thing you want to do is to form a carbon chain. Carbon chains form the skeletons of most organic molecules, and carbon chains may vary in length and shape. Let's take a look at length. Here we see ethane again, a 2-carbon molecule. Next to it is propane, a 3-carbon molecule. Octane makes up the fuel for your car tank, and that is an 8-carbon chain molecule. Chains may also contain branching off the main chain. Butane is a 4-carbon molecule. We can rearrange the butane molecule by removing one of the main chain carbons and replacing it onto the central carbon. This creates an entirely new molecule called 2-methylpropane or isobutane. Note that both of these molecules have the molecular formula C4H10. They are called isomers. We will revisit this shortly. Now let's look at double bond position. We were introduced to double bonds earlier with ethane and ethene. When we take the butane molecule present in box B and introduce a double bond, we produce butene. We have two options of where to introduce the double bond, either between the first and second carbon or the second and third carbon. Notice how if you placed it between the third and fourth carbon, you'd actually be creating the same molecule as when you place it between carbon 1 and carbon 2. Also notice that once we introduce a double bond, we needed to remove some of the hydrogens so that we always satisfy the octet rule. Carbons must always have 4 bonds, and hydrogen must always have 1. You will learn more about this when you get to organic chemistry. Lastly is the presence of rings. We can always take our carbon chain and form a ring. The smallest possible is 3-chain ring, but it's highly unstable and not often found in nature. The most common ring is the 6-carbon hexagon. Nature loves this shape. You find it in honeycombs as well. A 6-carbon ring with all single bonds is called cyclohexane. A 6-carbon ring with 3 single bonds and 3 double bonds alternating has a special name. It is called a benzene ring. As you progress through the health sciences, you will recognize this molecular structure on amino acids, pharmaceuticals, and lab chemicals, the benzene ring. All of the molecules we just learned have one thing in common. They're all solely made of carbon and hydrogen, no other element. Such molecules are referred to as hydrocarbons. Notice even the word hydrocarbon contains hydrogen and carbon. Hydrocarbons are found in organic molecules such as fat, as well as fuels like the octane and propane molecules we saw earlier. A major characteristic of hydrocarbons is their ability to undergo reactions that release a large amount of energy. Take a moment to consider the hydrocarbons I just mentioned, fats and fuels. Our bodies metabolize fats for energy, and our cars metabolize octane fuel for energy. Here's an example of the fat molecule our bodies metabolize for energy, a triglyceride or triacylglycerol. In future chapters, we'll dive into the types of lipids and learn more about triglycerides. For now, notice the structure of the triglyceride. There are 3 chains attached to a core. The 3 chains are called fatty acids and are completely comprised of hydrogen and carbon. They are hydrocarbons. We store triglycerides in specialized cells called adipocytes. Adipocytes are easily recognized under the microscope because they contain large fat droplets that are made up of triglycerides. The human body is adept at storing energy for times when there may not have been sufficient food sources. This is an evolutionary adaptation. It may not be as essential now as it was for our ancestors, but it is still part of our genetic makeup. I mentioned the term isomer earlier. Isomers are compounds with the same molecular formula but different structures and properties. There are 3 types of isomers: structural, cis-trans, and enantiomers. Structural isomers are molecules that have different covalent arrangements of their atoms. You've already seen structural isomers. In this example, we have pentane, a 5-carbon molecule with a molecular formula C5H12. Next to it is another 5-carbon molecule with the very same molecular formula, C5H12. What's the difference? The molecules have different covalent arrangements of their atoms. These 2 molecules will also have different chemical properties. Cis-trans isomers are molecules that have the same covalent bonds but differ in their spatial arrangements. Usually you will find cis-trans isomers relating to double bonds. The cis isomer on the left contains the 2 X's on the same side of the double bond, while the trans isomer on the right contains the 2 X's on opposite sides of the double bond. Enantiomers are isomers that are mirror images of each other. The left-handed molecule is called the L isomer and the right-handed molecule is called the D isomer. These might seem like minor differences, however, right and left-handed enantiomers have vastly differing effects in the body. Enantiomers are especially important in the pharmaceutical industry, where 2 enantiomers of a drug may have differing effects. Usually, only 1 enantiomer is biologically active. This slide shows 2 pharmaceutical drugs, ibuprofen and albuterol. Ibuprofen is a generic name for Advil. Only S-ibuprofen is effective here. This demonstrates the sensitivity of organisms to even subtle variations in molecules. Remember, structure and function are closely tied in biology. The remainder of this chapter will focus on special groupings of molecules called functional groups. Each functional group has distinctive properties and are key to molecular function. These functional groups are found attached to the carbon skeleton backbones of organic molecules. Here is an example of how adding different functional groups to the same base molecule will result in vastly different molecular functions in the body. Pictured are the molecular structures of 2 important hormone molecules for female and male development. On the left we have estradiol, a form of estrogen. Estrogen is paramount in reproductive regulation for females. On the right we have testosterone, the hormone responsible for development of masculine traits. Notice the base structure is largely the same, a 4-ringed steroid. We will learn more of this structure in a later chapter. For simplicity, molecular structures are often drawn without showing the carbons in the carbon skeleton chains. You can assume that every corner of the hexagonal and pentagonal structures contain a carbon atom. Now pay attention to the functional groups. Estradiol and testosterone have 2 functional groups in common at the top right of the structure, a CH3 called a methyl and an OH called a hydroxyl. However, in the bottom left of the structure is where they differ. Estradiol contains a lone hydroxyl group, OH, while testosterone contains a methyl, CH3, and a carbonyl, an oxygen with a double bond. Seemingly small differences lead to vastly differing molecular functions in the body. This is the role of functional groups. Functional groups are the components of organic molecules that are most commonly involved in chemical reactions. The number and arrangement of functional groups give each molecule its unique properties. You will learn the 7 most important functional groups: hydroxyl group, carbonyl group, carboxyl group, amino group, sulfhydryl group, phosphate group, and methyl group. Group number 1, the hydroxyl group. Hydroxyl groups are comprised of an oxygen attached to a hydrogen. The single solid line emanating from the oxygen indicates a covalent bond. Remember, oxygen has a valence of 2. The compound name for a molecule containing a hydroxyl group is an alcohol. You may associate the word alcohol with one particular substance, but it actually comprises an entire group of molecules. An example of an alcohol is an ethanol. Notice its similarity to the molecule ethane we discussed earlier. Ethanol is ethane with a hydroxyl attached to it. Ethanol is also called ethyl alcohol. Notice all atoms have the appropriate bonds to fulfill their valences. Carbon has 4, oxygen has 2, and hydrogen has 1. Hydroxyls have their own set of properties. Remember, oxygen is a very electronegative atom, meaning it has a high affinity for electrons. In covalent bonds, they hog the electrons that are being shared, creating a polar covalent bond. Therefore, this functional group is also polar. Polarity due to electronegativity creates partial polar charges. Oxygen will have a partial negative charge and can therefore create hydrogen bonds with any surrounding water molecules present. Water is, of course, a polar molecule itself. Group number 2, the carbonyl group. Carbonyls are comprised of an oxygen double bonded to a carbon. The carbon can then have 2 additional bonds emanating. 2 different compounds are formed depending on what is attached to that carbonyl carbon. If the carbonyl carbon is flanked by 2 additional carbons, that compound is called a ketone. If the carbonyl carbon is flanked by 1 carbon and 1 hydrogen, that compound is called an aldehyde. The simplest ketone is an acetone. This is a substance often used in nail salons to remove polish. You may recognize other ketones by paying attention to chemical names. Ketones often end in -one. For example, testosterone, lactone, and acetone. An example of an aldehyde is propanal. You'll notice that this is a 3-carbon propane molecule, just like the one we saw earlier, except one of the hydrogens is replaced by a carbonyl group. The ending -al indicates an aldehyde. Sometimes the word aldehyde is even part of the name, such as in formaldehyde. We also find ketones and aldehydes on sugars. They are called ketosis and aldosis, respectively, when they're on the sugar. We'll see more of this in the next chapter. The more you encounter molecular structures, you'll begin to familiarize yourself with the classification and the naming conventions, and when you take organic chemistry, you'll get a proper introduction to these conventions. Group number 3, the carboxyl group. Our carboxyl group is similar to the carbonyl group that we just learned about. In fact, carboxyls contain carbonyls. However, carboxyl carbons all contain an OH hydroxyl group on one side. The other side is usually attached to another carbon. Carboxyls act as acids. Recall from chapter 3 that acids donate H+ hydrogen ions, which are essentially a single proton, to solutions. More H+ in a solution leads to a decrease in pH and an increase in acidity. The carboxyl shown here is acetic acid, which is in vinegar. Acetic acid is a weak acid that can release its hydrogen proton under certain acidic conditions. Release of the proton creates the ionized form of COOH, which becomes COO-. Other examples of carboxylic acids would be lactic acid, pyruvic acid, and citric acid, which we will see in the citric acid cycle. Group number 4, the amino group. An amino group is comprised of a nitrogen attached to 2 hydrogens. Remember, nitrogen has a valence of 3. It wants to make 3 covalent bonds to satisfy the octet rule. In the previous slide, we saw what makes up organic acids. On this slide, we see what makes up a base. Recall from chapter 3 that bases accept H+ atoms in solution. Less H+ in solution leads to an increase in pH and a decrease in acidity. Make sure you understand this concept of acids and bases. Now, look carefully at the example of the base glycine. Glycine is one of the 20 amino acids, the building blocks of proteins. The amino group on the left side of the equation begins with a nitrogen covalently bonded to 2 hydrogens and a carbon. This satisfies its octet rule. The power of the amino group is its ability to take on an additional H+ proton. We then see on the right side of the equation a nitrogen with 4 covalent bonds. This is more than its valence of 3. It also has a charge now because it accepted the proton, which carries the 1+ charge. The amino base begins as NH2, and once it accepts the hydrogen proton, becomes NH3+. Such compounds are called amines. Group number 5, the sulfhydryl group. A sulfhydryl is comprised of a sulfur atom and a hydrogen atom. Sulfur plus hydrogen yields sulfhydryl. Molecules carrying the sulfhydryl functional group are called thiols. An example of a thiol is the amino acid cysteine. Cysteine is one of the 20 amino acids used to build proteins. One very important role of the sulfhydryl group of cysteine is to create cross-link bridges that help to stabilize proteins. We will see more of this in the coming chapters. Group number 6, the phosphate group. The phosphate group is comprised of phosphorus atom surrounded by 4 oxygens. Phosphate's valence is 5, and you will notice 5 covalent bonds emanating from the central phosphorus. You will also notice the 2- charge on the phosphate group. Why do you think this is? Remember, oxygens are highly electronegative and therefore have a high affinity for electrons. These phosphate groups actually begin with hydrogens attached. However, due to the high electronegativity of the phosphate group, the oxygens hog the electrons and release the H+ proton into solution, leaving 2 oxygens with negative charges. Phosphate groups are found throughout the body. Some notable places are within the backbone of all DNA and RNA, as a part of the energy molecule ATP, and phosphates have a special role in activating and deactivating molecules. You will learn more about this in your biochemistry courses. Group number 7, the methyl group. Methyl groups are comprised of a central carbon with 3 hydrogens attached. These are fairly small functional groups and are found on many organic molecules. The example shown is 5-methylcytosine, a modified component of DNA. Methyl groups are used in gene expression. They can be added to or removed from a part of the DNA to activate or deactivate a part of the genome. We call this unique ability to regulate gene expression epigenetics. These compounds are then said to be methylated. To finish this chapter, we are introduced to the energy molecule of the body, ATP, or adenosine triphosphate. ATP stores the potential to react with water. Such a reaction results in the release of energy, which can be harnessed by the cell to do work. Note the structure of adenosine triphosphate in the simplified diagram below. We have the adenosine molecule attached to 3 phosphate groups. After the reaction with water, ADP is released, which is adenosine diphosphate, as well as the inorganic phosphate group we've just cleaved off of ATP. Energy is also shown released here. We will discover how the body harnesses the power of ATP hydrolysis in a later chapter. Be sure to attempt the concept check problems presented at the end of this PowerPoint presentation. Next chapter, we will discuss the structure and function of large biological molecules.